Good wishes. I'm Kumar here. I'm a genetic counselor from India, and we do genetic counseling for patients with various genetic disorders, both online and offline. Today, I'm going to discuss about Usher syndrome and give you an update on the genetics, genetic counseling, and therapies. My talk is divided into three segments. First, I'll explain what is Usher syndrome, and then we will discuss how patients come here for genetic counseling with what needs. And then we look at current therapies, which are both in the preclinical research as well as in the clinical trials. The way we sense the external world is through various sense organs in our body. Starting with skin, we can feel the touch, the pressure, and the temperature through various nerve endings, which we call as receptors. Through the tongue, we can taste and then have a good meal and enjoy it. Through the eyes, we see the light and the vision and perceive it through various sensory receptors in the eye. Through the ears, we can hear the sound, and also the inner ear helps us in balance. As far as the nose is concerned, it helps us to smell the external world. Besides these, we also have one more sensation called as vibration. With these various sensations, we perceive the external world and then accordingly we respond to it. These sensory end organs, which we call as sensory receptors, are of different types depending on which part of the body they are in. As far as the Usher syndrome is concerned, with the retina in the eye and the cochlea in the ear, we have sensory receptors in the retina called as rods and cones. In the ear, we have the sensory receptor for sound. The human eye is actually a window to the world, and 80% of all the sensation is through the vision that we enjoy. We can perceive 10 million different types of colors, and the human eye resolution is about 576 megapixels, and no camera can actually match that today. Compared to the eye and the ear, the eye has certain muscles around it, about 6 of them to each eye, and that helps us to see the world with 180 degrees. If you see the parts of the eye and through a cut section, the first part is actually by the cornea, which is transparent, and behind that we have the iris, which forms the pupil or the aperture of the eye. And behind the pupil, you have the lens, which helps us to focus whatever object that we may see. And all these are in the front part of the eye, which is called as the anterior segment. And in the posterior segment or back of the eye, we have the retina. And the retina has these photoreceptors or sensory organs of the retina to perceive light and vision. And these nerves finally join together and form the optic nerve, which goes to the brain. And what you see in this image is an electron microscope photograph of the rods and cones. And these rods and cones join together to form the optic nerve, and they go to the brain. To the left, you see the eye of human on the top and that of the mouse below. And you can see the difference between these two different types of species eye. And the retina, if you actually really want to imagine how it will be if you break an egg and the shell, and adhering to the shell is a transparent or translucent membrane, and it is dirty white in color. That is the thickness of our retina. But nevertheless, it has 10 different layers, starting with the photoreceptors of rods and cones. And all these other 9 layers process the visual sensation and then send them to the brain through the optic nerve. To the right, you see the rod and the cone. And the rod helps us to see when the light is less or dim light. And the cones help us to see when the light is bright. And cones also help us to read and appreciate colors. So, as mentioned, the rods are sensitive for black and white color, and they work in dim light. And cones are responsible for color vision, daylight vision, as well as reading ability. Each of our human eye has about 120 million rods and 6 to 7 million cones, and all of them reach the brain. If you see the other component of Usher syndrome, which is hearing, the hearing is facilitated by the ear, and we have the external part called as the pinna, which is a cartilage. And then we have the ear canal, which ends with the tympanic membrane. And behind the tympanic membrane, or the eardrum, we have 3 different bones called as malleus, incus, and stapes. And the stapes is attached to the cochlea or the inner ear. And when sound waves come, the tympanic membrane vibrates, and that will make the 3 different bones to move. And that movement will create the stapes to move the fluid inside the cochlea. And when the cochlear fluid moves, then the hair cell in the cochlea will actually move and make us feel the sensation of sound as you're hearing me talk. There's also another component of the inner ear, which are semicircular canals, and they will help us to have balance. So the nerves which start from retina or from the cochlea will end up in the brain in 2 different parts. The visual pathway will end up in the occipital lobe, exactly behind our brain. And the cochlear signal reaches the temporal lobe. So the ear and the eye are tools for the sensation of vision and hearing. So the perception of vision and hearing is actually processed in the brain. So eye and ear are tools, and so are the other sensory organs in the body. Now, if you compare vision and hearing to a car, and you want to start and move it, you put the key in the ignition, and then there will be sparks that comes out of the spark plug in the engine, and that spark will ignite the gasoline, and the gasoline will try to expand and cause a combustion, which will move the pistons. And when the pistons move, then it actually moves the axle in the car, and the axle will move the wheels and the tire when we press the accelerator pedal. Okay, so the minute you put the key and start the engine, till the car moves, there could be a couple of hundred reactions that takes place in the car. Similarly, when the light, the photon, strikes the retina, there are a couple of hundred reactions that takes place, which finally becomes a visual signal that reaches the brain. And so does it happen in hearing when these signals reach the temporal lobe in the brain. And therefore, we perceive vision and hearing. Now, in the retina and in the ear, there could be a couple of hundred genes which produce these proteins, which could be structural, physiological, and biochemical in nature. And all these proteins produced by the genes will finally end up in a symphony which we call as hearing and vision. So one of these genes, of a couple of hundred in vision or hearing, if they don't function properly, because there is a DNA letter fault, or which we call as DNA variant or mutation, then that results in the individual's inability to perceive vision or hearing. So all these genes have to function in coordination, and so will the proteins in the retina and in the cornea. Now, in Usher syndrome, as I proceed with my talk, you could take a photograph of the retina of the individual, and this is how the retina will look like. I'm not an ophthalmologist, I'm a geneticist. So what you see here is the pigments in the retina of the individual. So it's a black and white photograph. This is a color photograph of the same individual. And you see that the blood vessels are thinned out, which we call as attenuation of the arteries. All this will lead to the photoreceptors dysfunction because one gene has a mutation out of a couple of hundreds. Then that will make the retina to degenerate and become dysfunctional. And start with different types of retinitis pigmentosa will have different features. So the one with the rod cone degeneration will start with night blindness and then it will lead to reading disabilities. Cone rod dystrophy will be exactly opposite. It will start with reading disability, color vision disability, and then it will move on to the night vision issues. So what you see here is that is of a patient who has Usher syndrome in a very advanced stage, but he has only retinitis pigmentosa. Then how did you actually discover that he has Usher syndrome? It is because we did a genetic test and he had a mutation in one of the Usher's gene. So it has been reported that people with Usher syndrome gene would have only retinitis pigmentosa and may not have a hearing issue. But majority of patients with Usher syndrome will have vision and hearing issues. So this is a photograph of the OCT image, and with which we can see the thickness of the retina and see how much of photoreceptor layer, the first layer of the 10 layers, is intact. What you see here is a pedigree of this patient, who is quite a senior man, and he's affected. His wife is normal, so we draw in the pedigree with a square for a male and a round for a female. And he has 4 children. The first child, who's a female, has the same problem, and she's around in her 40s as far as her age is concerned. The other 3 children, a male and 2 females, do not have vision or hearing problem. So therefore, we take a detailed pedigree of the individuals. I'm not showing the detailed pedigree for privacy reasons of the individual. I'm showing a very brief shorthand of the pedigree. But Usher syndrome is mostly an autosomal recessive condition, okay, where both the genes that we inherit from our parents are defective, and that leads to the vision and hearing problems in Usher syndrome. For various genetic reasons, which I cannot explain here, you have both the father and the daughter having the same condition. Now, Usher syndrome has a lot of genes, about a dozen of them, which, if mutated, results in Usher syndrome. And in an individual, only one of these genes could have a defect, and that will result in Usher syndrome. Now, Usher syndrome has three different clinical types: type 1, type 2, and type 3. And in type 1, they're born with profound hearing loss, and they will also have vision issues, which are very progressive. And they would have, mild balance issues, and mostly these balance issues, if you take 100 patients with Usher's, very few of them have a balance issue. In type 2, they're born with mild to severe hearing loss, and it could be progressive, and they could have a vision issue and no balance problems. In type 3, they're born with normal hearing, and there could be a progressive hearing loss, and they will have the vision related retinitis pigmentosa, and they could have some balance issues. So each of these types have different genes which are responsible for the disease type. And doing a genetic test will help to know which type it is. And some of these genes are in gene therapy clinical trials, for example, USH2A. So why does a fault in a gene cause both hearing as well as vision issues? Therefore, since it is present in more than one part of the body, we call it as a syndrome, and therefore Usher syndrome. The Usher syndrome gene, which produces a protein, is like a cilia. Okay, and therefore it is a ciliary protein. It is present in the photoreceptor as well as in the receptor for hearing. And therefore, when you have a mutation in one of these genes, it will result in both the receptors in the retina and the cochlea damaged and affected, which leads to vision and hearing problems. Now, when do we do genetic counseling? Every patient with a genetic disorder does not need genetic counseling, but there are only certain circumstances in which a clinician or a patient can come for genetic counseling. The clinician can refer the patient, and the patient themselves can also come for and take the counseling session with the genetic disorder. There are only 4 reasons. I'm not showing the families because this is a patient group, and for privacy reasons, I'm not showing individual cases. But in our practice, we have found only for 4 reasons they come to us for genetic counseling, or the clinician refers them for genetic counseling. The first is where there is ambiguity in clinical diagnosis. Okay, so when you do a genetic test, you know the gene, and then you can actually know what the condition is. As I told you in an earlier case that it was a retinitis pigmentosa without any hearing loss, but when we did a genetic test, we found that the person had an USH2A gene variant. Now, the genetic inheritances can be of different types. As you can see here, one is called as autosomal dominant, other is autosomal recessive, third is X-linked recessive, and fourth is mitochondrial. A few other genetic types which are quite uncommon and not mentioning it here. In autosomal dominant, there's only one gene defective compared to the other normal gene. Okay, so we get almost a pair of genes from our parents, and each of that gene will do its function. And in autosomal dominant, one gene will be defective and the other gene from one of the parent will be normal. But despite having a normal gene, the defective gene will dominate the normal gene, and therefore it is called autosomal dominant. And in this condition, the age of onset or the severity of the disease will all be much milder. So it will be late onset and it will be milder and the vision will be retained for a long period of time. In autosomal recessive, both the copies from the parent can be defective and that could lead to this autosomal recessive condition, which will be early onset and severe. In X-linked recessive, there is a variation between genders. So a male will have one X chromosome, the other chromosome will be Y chromosome, whereas females will have two X chromosomes. And therefore, if one of the gene is defective, the male is affected because he doesn't have a compensatory X chromosome. So this other chromosome is Y chromosome in the male. So females will be carriers of the disease and not affected, and males will be affected with the condition, and it will be like autosomal recessive, early in onset and quite severe. Mitochondrial gene defects can be in the mitochondria, and therefore it will be early onset and very severe. So knowing the genetic type will help us to counsel and tell about the progression of the disease depending on what genetic type it is. So identifying syndromes, it'll be very useful if you do a genetic type, genetic screening and come to the clinical diagnosis. So that's the first column. The second reason why they come is they want to get married. Somebody is affected, wants to get married, wants to know if the child will be affected. The second situation where somebody in the family is affected and therefore they want to know if they will have similar kind of children. And therefore, for a reproductive choice, they come for the premarital genetic counseling, as you can see in the second column. The third column is when there is a family history, particularly first child is affected with a genetic disorder, then they will want to know what are the chances for next child, which we call as preconceptional genetic counseling. And they could also go for a prenatal genetic counseling where a prenatal test is done by taking a sample even before the child is born from the placenta of the mother or from the amniotic fluid. Once we know the child has the genetic defect or does not have the genetic defect, then the parents can choose themselves and decide what they want to do with the pregnancy, whether to go for an abortion or to go ahead with the pregnancy. Now, when you have an issue with abortion for various reasons, social reasons or religious reasons, then you could go for something called as pre-implantation genetic counseling, where in a test tube embryo, you can test and see if it is carrying the defect, which we call as in vitro fertilization and embryo transfer. So the embryo can be tested, and the ones which do not have the mutation can be implanted or embryo transfer. All this is possible in most parts of the world today. Now, the fourth category for which they come is when they come to know about various therapies that are available, and they want to know if they can be a candidate for that. So, various interventional strategies are there, about which I'll talk subsequently. And therefore, it can be in a preclinical state where there can be cell models, animal models in which scientists will be doing research. And it would have passed that preclinical stage and would have gone to clinical trials. And USH2A is in clinical trials, particularly one type which I will mention later. And there are certain genes that have come into therapeutic options. One amongst them is RP65. The Spark company, the US, is actually providing this. Okay, what are the therapies that are available today? I've also given some references if somebody wants to get more information. Now, Usher syndrome, you can have a gene defect and a mutation producing an altered or non-functional protein. And you can change that and put a new gene in the individual with the help of a virus, which we call as gene replacement therapy. There's another methodology called as genome editing, where we can correct the DNA letter defect. You can also intervene with RNA interventions like antisense oligonucleotide RNA intervention or translation read through drugs, again, which are RNA interventions. So the H2A, which a company called Sepal Bio is doing, is present in the US as well as in Europe. And they try to treat people with Usher syndrome who have a change in the middle of the protein in a part called exon 3. So a gene has compartments, and some of the compartments are translated into protein, and some of the compartments are not. The ones that are translated are called exons. So individuals who have a variant in exon 3 can be treated by this exon skipping method, where the methodology will go to the 12th exon, and from the 12th exon, you jump to the 14th and not translate the 13th. So this is called as exon skipping method. Now, besides these, you have cell therapies, particularly with stem cell. And all this research are progressing very well. And there is a lot of hope that people who are listening to my talk can be hopeful that there will be a treatment sometime in future. It's difficult to tell the time, but certainly the way things are going today in therapy research for genetic disorders, a lot of promise is there. So with that, I come to the end of my talk on Usher syndrome. First, I explained what is Usher syndrome, which is a problem that affects the receptors in the retina as well as in the cochlea. These are sensory receptors. In genetic counseling, people come for 4 different reasons with our experience. One for clinical diagnosis. Second is for premarital counseling. Third is for preconceptional or for prenatal genetic counseling. And fourth is when intervention strategies are there. Some of the patients want to know if they will be eligible for that in future. I spoke about therapies and a lot of promises there. And if you really see what is the big challenge that is there in this field, I would like to quote another situation. The entire world was under the pandemic, and to find a vaccine, it took only 9 months. Okay, the technology was there, the clinicians were there, the scientists were there, and the big challenge was the funding. So once funding is available in record time, like the way vaccines were produced for the COVID pandemic, you can actually get things done. So there will be certain, mistakes, errors that a scientist will come across and the clinician will come across, and they will correct it. So that is why things take a long time. But once they correct, they need further money and funding that they need to take things forward. So when patients groups and parent groups put their mind together and find the money, then certainly all these conditions time will come when therapies will be available. With that, I almost come to the end of my talk, but then I have to thank people who were able to make me give this talk. And what you see here is Eye Hospital, Narayan Bangalore. In the top, you see the clinicians, okay, both the ophthalmologist, all of them are ophthalmologists. And in the bottom right, what you see is the scientists who are working in the laboratory. And putting things in one line, we are fighting blindness and finding solutions to cure people with various visual disabilities. I'd like to thank the Usher Syndrome Coalition Group for giving me this opportunity. And also I'd like to mention in Bangalore, Anuprita Ghosh helps me in genetic counseling. This is one city in India called Bangalore, and there's another city in India called Chennai, where also I work. And to the left, you see three different ophthalmologists, clinicians who are working on their research topic and doing PhD. They're qualified ophthalmologists practicing ophthalmology, and they're surgeons too. And they are working on a research topic and doing PhD. It's very important for clinicians to do science and try to understand the fundamental basis of various diseases so that we can find out interventional strategies. To the right, you see mostly the master's students who are doing PhD. Brinda does help me in genetic counseling in Chennai, and then you have Deepika and Sivita. And Dr. Rajeshwari is a pediatrician, and she's yet to register for PhD. And we do this PhD program with the Maastricht University in Netherlands. With that, I come to the final slide of my talk, where I spoke about Usher syndrome and gave an update on the genetics, genetic counseling, and therapies. Thank you.